right, now we get to telophase and cytokinesis. So in telophase, you start to see sort of a squeezing of the cell membrane starting to occur here. The chromosomes have moved all the way in, and you start to see the nuclear membrane start to reform. And you start to see a dissociation of the chromosomes. They start to uncondense. And then you have actin and myosin playing a role here in constricting down the cell membrane to separate the cytoplasm. So here in telophase, we have cytokinesis at the end of this occur, where you separate out the cytoplasm. Now the mitochondria that are in here, the thing to note about mitochondria, they also go through a division as well, but they don't necessarily follow through with the division during the actual mitosis of the actual eukaryotic cell. They'll divide on their own time, but they will get divided up between the cytoplasms. They'll be separated out from each other during this constriction. In animals, we have the formation of what we call the cleavage furrow. In plants, we have the formation of a cell plate. So you start to lay down the cellulose between the two. So telophase and cytokinesis. Telophase is the stage, cytokinesis is the separation of the cytoplasm. The DNA has already been separated. That happened during the metaphase, the prophase on up to anaphase. That's called karyokinesis. That's the separation of the DNA. We can actually visually see these on fertilized eggs. We call them cleavage furrows. It's just a cleavage of it. Now in eggs, they don't really go through a G1 or a G2. They just go through S and mitosis. But you can see this looks like, you can see how the actual membrane is being constricted down. This is using actin and myosin to do that constriction. Whereas in plants, you actually see a cell plate being laid down. So you have a cellulose being laid down between the two sets of DNA and cytoplasms, as well as a membrane being laid down. So the outcome of mitosis for it. Basically, you have mitosis and cytokinesis produces two daughter cells. They have the same number and complement of chromosomes as the mother cell. Okay? They're genetically identical to each other, barring any rare mutations. They ensure basically that you have genetic consistency from one cell to the next. So there's not a mixing of DNA, it's just a replication of the DNA and then a separation. So they are genetically identical. As I said, sometimes there's rare mutations occur. We'll study those in a different chapter. The development of multicellularity relies on the repeated process of mitosis and cytokinesis and the fidelity of that it does not have mutations. That's how we came about from a single cell, is through the mitosis. Okay? Plus you have an equal distribution of genetic material and no loss of genetic material to bring about this body. Now let's get to meiosis, the production of new individuals, progenies, for multicellularity. Okay? So we can have meiosis also in yeast cells too. They go from a haploid to a diploid, and then they do meiosis in response to environmental changes. Sexual reproduction is a common way for eukaryotic organisms to produce offsprings. So the parents are going to be diploid, and then they produce gametes that are haploid, or spores in the case of plants that are haploid. They will fuse together each other during the fertilization to create a new diploid individual. So this is a mixing of the DNA between two different individuals. Their DNA are going to be different, and so this is when you start to see the expression of those genes in relationship to each other and the environment around them. Meiosis produces haploid gametes. They contain a single set of chromosomes. Basically, a diploid human cell contains 46 chromosomes, whereas the gametes are going to contain 23 chromosomes. So one gamete is going, if the sperm is going to be the deciding factor whether male or female. The female egg only has an X chromosome for the sex chromosome, whereas a male can have either a X chromosome for one sperm or a Y chromosome for the other one. So you have 23 chromosomes. During meiosis, haploid cells are going to be produced from the diploid cells. Thus, chromosomes must be correctly sorted and distributed to reduce the chromosome number to half its original value. You can't have a problem there. You need to have good segregation of the chromosomes. So that has to happen in order to get it to bring them back together to have proper development. In humans, for example, a gamete must receive one chromosome from each of the 23 pairs. Now we have some stages that are built into meiosis that are different. We'll actually go through 2 rounds of karyokinesis. in meiosis. So first round is going to be called meiosis 1. In this one we have prophase 1, and you'll notice there are 5 different stages associated with prophase 1, and there's a reason we're going to talk about this, is because this has a lot to do, especially when we talk about mammals. In the female stage, the actual eggs are going to stop at one of these stages and wait for fertilization before continuing with these stages. And we see this happen in that systems. So, and it differs between the different mammals. So this is actually the separation of the homologous chromosomes, okay? So remember, in humans you're going to have 4 sister chromatids for each chromosome set. And then what's going to happen is, to set this up, you're going to have those dyads are going to line up and they're going to separate during meiosis 1. So you have 2 of the sister chromosomes over here, 2 sister chromosomes over here, they separate from each other. And they do this, they go through metaphase 1, anaphase 1, and then telophase. Then they go into meiosis 2. Notice we don't have these stages over here in this, and that should be prophase 2, by the way. And then over here we don't have those particular stages in this system. So we have metaphase 2 after that, anaphase 2, and then telophase. So there is a division down here, a cytokinesis here and a cytokinesis here. So stages of prophase of meiosis 1. We call this leptonine. Leptonine is going to be where the replicated chromosomes are going to be released. They're going to have basically you're going to have the system set up to where the chromosomes are going to come together. So if you'll note here, here's the 2 types of chromosomes here. These are, just looking at these, so this might be chromosome number 1. They're going to start to come together with each other during leptonine. Nuclear membranes here. Zygotine, bivalent formation. This is a bivalent structure. These guys are going to fuse together. It's called the synaptimal complex is going to form here. So they're going to fuse together. And then by pachytene, what will happen because of the fusion, there are going to be some enzymes that are going to go up and down this synaptimal complex and choose DNA from the 2 different chromosomes and actually cross them over. They're going to take this DNA from this chromosome, this one, and separate them out. This is called crossing over. And it was defined early on in the study of the genetic material as a way to explain why they thought that the DNA was being mixed the way it was. And it turns out it's true. By diplotin, you have these things lined up. The synaptimal complex starts to dissociate so that we can go into the anaphase for this. So then they separate out. You have the end of prophase 1. In the metaphase. So we can go into metaphase and then anaphase. So this is the stages, and what we'll see happening is that at this particular stage here in humans, you have the egg is kind of locked at this point until it's released once a month, okay, during the menstrual cycle. So it's going to be locked at this particular point here, and then it's going to be released from that, and it's going to continue with the prophase and finish it off. So meiosis 1, we have early prophase, late prophase, and then prometaphase. So the 5 stages that I just talked about: pachytene, the leptotine, zygotine, pachytene, diplotin, and then finally dikinesis, where they pull apart and you can see the actual crossover structures. And so we're going to go into metaphase, anaphase, and then telophase and cytokinesis. Now meiosis 2. In meiosis 2, we've already separated the homologous chromosomes. That's what happened here. We separated out the homologous chromosomes. Notice how they still have an X shape to them. So they still have a sister chromatid. This one is homologous to that one. That one's homologous to that one. Notice how the DNA has been exchanged here. So this is one dyad and this is the other dyad. So the homologous dyads have been separated from each other. Now we have to separate the sister chromatids. We still are 2 in at this stage. Now we have to go down to 1 in. So you have 2 of these. They're going to go through prometaphase, getting ready to line up the attachments. They're going to line up at metaphase. We're not going through the 5 stages of prophase as we saw in meiosis 1. They don't need to. Here, now we have a monad. Notice one sister chromatid. They've been separated. So the centromeres have separated at this point, and they go to their respective particular cell. And these are going to be haploid cells. That's the gametes. And that's the concept of meiosis 2. So if we look at the comparisons, here's mitosis. This, remember, it goes 2 in to 2 in. Here we're going from 2N to 1N. And so we don't have all these various stages between middle prophase, late prophase, the pachytene and all of that, leptotene. You don't have those up here, but we do have them here because we have to line up the homologous dyads so that they come together, creating what we call a tetrad. They line up, see how they're lined up here versus lined up here for mitosis? So the homologous chromosomes are lined up, crossing over can occur, and now they go to the next stage where, in this case, they go to anaphase, telophase, and they have 2N cells, but here they have to go through a second round of meiosis, cell division, they separate out, notice there's no more crossing over occurring, and now we have 4 gametes versus 2 daughter cells up here. So what's the significance of meiosis? After a cycle of meiosis, well, let me start over. We'll stop at that point.